The overall goal of this experiment is to determine the absolute protein copy number of a target protein with single-cell resolution.
Single-cell techniques will enable a better understanding of the behavior of a protein as it relates to the central dogma, and help toward accurate quantitation of the control of gene expression. Working at the single-cell level can capture the rich heterogeneity of a cell population that would otherwise be lost by the averaging that occurs with traditional biochemical techniques.
Generally, individuals new to this method will struggle because of the required sensitivity of single-cell assays, owing to the limited amount of protein in each cell.
We first had the idea for this method when we wished to perform a single-cell protein assay that could be easily sampled for further analysis downstream.
Begin this procedure by making chips and printing antibody microarrays as described in the text protocol. To prepare the apparatus, feed a 100 mm length of 150 micron inner diameter, 360 micron outer diameter fused silica tubing through a 40 mm length of 1 mm inner diameter, 1/16 inch outer diameter PFA tubing until it protrudes by 2 mm. This will form the concentric nozzle. Apply a thin layer of cyanoacrylate glue to the end of a 10 microliter pipette tip and insert it into the 40 mm piece of PFA tubing. If required, reposition the fused silica tubing to maintain a 2 mm protrusion at the nozzle before the adhesive sets. Insert the other end of the fused silica capillary into the end of a 200 mm length of 0.014 inch inner diameter, 0.062 inch outer diameter PTFE tubing. Connect this to a 100 microliter Hamilton syringe filled with 4% bovine serum albumin in phosphate buffered saline.